All right, let's begin. I was a little bit worried. The first time I tried to get into Zoom, it said you were not authorized to start the session or something like that. And it told me it timed out or something. And so I said, geez, that's weird. I'll just try it again. And fortunately, the second time it did work. I had to just go to my, what do you call it? The Canvas page and go to Zoom and click on the link in the Zoom page. Anyways, I'm here now. So are there any questions? No questions, so we'll begin. Let me share my screen. Oops, that's a little weird. Let's get this out of there. So today is February 2nd, and we're supposed to be doing Lab 6, disk diffusion and antibiotics. You don't really need to do anything further on your unknown project, except obviously read the material I went over and look through that material. And there's a lot of material to look through, but you can look up what the possible bacteria species are. Let me see if I can find that now. You can go look at, come on. Week 5 lab, I believe. You can look up the example of a lab report. That's the sample journal. The instructions, which hopefully you've already read. If you haven't, make sure you do read that. Building a dichotomous game plan, unless you know what doing the project dichotomously means, and you will only do it dichotomously. You could look at the unknown list. I already mentioned that, but look at the report grading. You don't really need to look at this one now, but before you write up your unknown report, you should look at this. Any questions about any of that? All right. I mentioned in the lecture that we'd done some grading, so I won't mention that here. And we don't have a quiz until next Wednesday. All right. If there's no questions, let's begin. So, Lab 6, disk diffusion. Let me blow this up a little. Evaluation of disinfectants and antiseptics and antibiotic effectiveness. We'll mostly be looking at antibiotic effectiveness, but the procedures given here can be used to evaluate disinfectants and antiseptics. So there's some reading to do for this lab, but not very much. Reading a page there, a couple of pages here on the principles of infective disinfection, a page there on chemical methods to microbial control, reading a little bit about, gosh, that's really only a page on antibiotics and taking a look at the table. And then test to guide chemotherapy, 2 pages there. There are some video clips today. I don't believe there is a virtual lab, but I can't remember which labs still have virtual labs and which ones don't. And I need to cross it out if I'm not using it. So let me see. Maybe I'll make it a different color and then we'll hopefully come back to that and take a look and see. I don't think we have a virtual lab today, but there are a few labs we have a virtual lab in, and I don't remember which ones. We had a whole bunch of virtual labs, and most of them use Flash, and we can't get them to run anymore because Flash is no longer supported. And when you try and get it to run, or you try to download it, Adobe will say, I'm sorry, you can't get it anymore. So we don't have as many virtual labs anymore. And surprisingly, I looked, because now it's been a year since Flash has not been used, and most people have not updated their Flash virtual labs and made other ones. And I looked for other virtual labs and I just couldn't find them. And like I said, it's been a year now since Flash has gone around, and I thought that there would be people who started making virtual labs and making them available, and I just couldn't find them. All right, primary learning objectives. Upon successful completion of this lab, you should be able to identify the type of chemical agent and the active ingredient used in the various disinfectants and antiseptics. You should be able to evaluate the effectiveness of disinfectants and or antiseptics using the disk diffusion method. You should be able to determine the mode of action of various antibiotics. You should be able to evaluate the effectiveness of various antibiotics using the disk diffusion method. You should be able to list the variables that influence the antibiotic disk diffusion susceptibility testing, meaning there are different things that can influence the results when you're running an antibiotic disk diffusion test. So list those variables. And then 5, be able to define the terms of this lab: sterilization, disinfection, what's a disinfectant, an antiseptic, a bactericidal agent, a bacteriostatic agent, an antibiotic, what's a spread plate, and then what's the zone of inhibition. And I usually will just abbreviate zone of inhibition as ZOI. So if ever I send you a note about that, ZOI means zone of inhibition. So sterilization is completely eliminating all viable organisms, including endospores. This is usually done by very high heat or autoclaving. Okay. And so we do not usually sterilize living tissue because the treatment would kill the living tissue. So like I said, you don't send a patient into the hospital for surgery and then sterilize them, because if you were to remove all microbes from their skin and inside them, you would kill the patient because it would the treatment would be too severe. Okay. Any questions about any of that? A disinfection is what we do to an object when we're destroying vegetative cells. When we disinfect an object, we do not necessarily kill endospores. Okay, any question about that? So disinfection is what you do to an object. We call it a disinfectant if it's a chemical agent used on an inanimate object to disinfect the object. On the other hand, we call it an antiseptic if it's a chemical agent and we apply it to living tissue. And that would be not called disinfection, that would be called, what is that called? Antiseption, something like that. To evaluate the effectiveness of chemical agents, the surface of a nutrient agar plate is inoculated with a particular bacterium, and usually the bacterium you want to test. Paper discs saturated with different chemical agents are then applied to the surface of the agar. The chemical agent will diffuse into the agar medium, establishing a concentration gradient around the disc. You then grow the bacteria on the plate overnight at 35 degrees and look at the growth pattern around the disc. If there is a zone of inhibition, an area of no bacterial growth around the disc, the chemical agent is assumed to be at least a little bit effective for that particular bacterium. Conversely, if there's no zone of inhibition, then you can say the chemical agent is ineffective against that particular bacterium. Now, this is a little bit of an oversimplification, and we'll talk about the oversimplification later in this lab module. But that's a quick way of looking at it. This table is telling you the different categories of chemical agents, like ethanol, isopropanol, they're an alcohol. Their general mode of action is protein denaturation and then dissolve the lipids. And their preferred use is an antiseptic, and they're really very weak antiseptics. So it'd be better to call them a degerming agent, because when we use them on our skin, for example, we just reduce the number of microbes in the area we degerm. We do not get rid of all of the bacteria or microbes in that area. And endospores in particular, does not state it here, would not be killed by alcohols. And then there's formaldehyde, formalin, glutaraldehyde, they're aldehydes. You can look at the general mode of action, the preferred use. We usually use these as a disinfectant, and these are for pretty good disinfectants. They actually do kill endospores. Halogens include things like chlorine bleach, and they're mostly used as a disinfectant, but they can be used as an antiseptic. And I think I've mentioned it used to be used that when somebody had necrotizing fasciitis, they would treat the skin of the patient with chlorine bleach. And that's because although chlorine bleach was irritating to the skin, it was preferred to the case of necrotizing fasciitis. And so they would actually use that as an antiseptic. I don't know if they still use that as an antiseptic, but at least they used to use it as an antiseptic for necrotizing fasciitis. All right, you can read about the other ones. I just want to talk about peroxygens. Hydrogen peroxide is a peroxygen, and it is used as an antiseptic or even as a disinfectant, and it is a very good antiseptic. Hydrogen peroxide can kill endospores. I don't know why that's not in the table there. That's one of the few agents that can actually kill endospores, although aldehydes can as well. Phenolics, used as antiseptic. When you use them in low concentration, they irritate the skin, so they're more likely to be used as a disinfectant. All right, any questions about any of that? The zone of inhibition size depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. Various other factors come into play and affect the zone size, including the concentration of the chemical. The more concentrated you have, generally the greater the zone of inhibition. The molecular weight of the agent, the larger the molecular weight, then the smaller the zone of inhibition, and then the density of the bacterial inoculum. The higher the bacteria, the smaller the zone of inhibition will be. So those are some of the factors that come into play for determining the zone of inhibition. In this lab, we'll mainly look at the evaluation of antibiotic effectiveness. And antibiotics are things we use against microbes, especially bacteria. Antibiotics are categorized as bactericidal if they kill the bacteria, and bacteriostatic if they inhibit the growth of the bacteria. When a bacteriostatic antibiotic is used, it will not remove the bacteria from the patient, but we're relying, or the doctors rely on the immune system to assist in eliminating the infection. And so usually a bacteriostatic antibiotic is effective because the host immune system will be given the time it needs to remove the bacteria from the patient. And the bacteria can't grow because of the bacteriostatic agent. The Kirby-Bauer disc diffusion test is the most common method used to determine antibiotic testing. The results of such testing help physicians in choosing which antibiotics to use when treating a sick patient. The bacteria are isolated from the patient and grown on an agar plate. Filter discs are impregnated with different antibiotics. And after you apply the bacteria on the plate, you let it sit for a few minutes, and then you put the filtered disc on the plate. The disc will establish an antibiotic gradient around the disc. You then incubate the plates for 18 hours at 35 degrees Celsius, and you look at the zone of inhibition, where the antibiotic concentration is high enough to inhibit the bacterial growth. The inhibition size is dependent on several factors. Obviously, it's dependent on how sensitive the bacteria is to the antibiotic. That's the one we're most interested in. But there are other factors that come into play too. The degree of sensitivity of the microorganism, the diffusion rate of the antibiotic is very important in determining the size of the zone of inhibition. The smaller the antibiotic, the greater it can diffuse on the agar. The larger the antibiotic, the smaller the size of the zone of inhibition. The growth rate of the bacteria also determine the size of the zone of inhibition. So you can determine that by plating a different concentration of bacteria, putting it on a different media. So because of all these variables that can affect the zone of inhibition when performing the Kirby-Bauer disc diffusion test, we standardize the Kirby-Bauer test, what procedures we use. First, we inoculate nutrient broth from several bacteria colonies that are isolated on a non-selective media. We then grow the bacteria in the broth for no more than 24 hours. So it's fresh growth. We then adjust the broth turbidity so that 0.5 McFarland units are used. And this will give you approximately 10 to the 5th microbes per mil. If you use a different concentration, you'll get different results. I'm just going to mention that. You then inoculate the bacteria onto a Mueller-Hinton agar plate. We always use Mueller-Hinton agar because different media can affect the test. You do it at room temperature, and the Mueller-Hinton plates are always pH 7.2 to pH 7.4. They always have a specific depth, 4 millimeters in size. And then we use a specific diameter of the plate, 150 millimeters size plate in its diameter. You put the bacteria on the plate with a non-toxic cotton swab using a spread plate technique to obtain confluent bacterial growth. And I guess I'll show that later, how we do that. You place the disc at least 24 millimeters apart and no more than 12 discs per the 150 millimeter Mueller-Hinton plate. So the discs have to be a certain length apart, and that's so that this one doesn't interfere with that one and vice versa. And then you add the antibody disc 5 minutes after you inoculate the plate with bacteria, but no longer than 15 minutes after you inoculate the plate with bacteria. you want to let the bacteria sit a little bit on the plate so that the liquids that you're putting the bacteria on are absorbed by the agar. And then you take the plate, flip it upside down, and incubate it in a non-CO2 incubator for 16 to 18 hours and at 35 degrees. And you put it in the incubator no more than 15 minutes after you add the antibiotic disc. So this procedure is highly standardized to make it so that wherever you are in the world, if two people are running the same Kirby-Bayer test on the same bacteria using the same antibiotics, that they will get similar, if not the same, results. That one person won't get large zone of inhibition and the other one will get a small zone of inhibition, etc. Okay, so they'll be similar. And that's the idea of standardizing the Kirby-Bayer susceptibility test. Any questions about any of that? All right. After incubation, the plates are taken out of the incubator and the inhibition size, the zone of inhibition size, is measured in its diameter using millimeters. The bacteria is considered to be sensitive to the antibiotic, intermediate, or resistant to that particular antibiotic, depending on the size of the zone of inhibition. And determine that, you have to look at the chart prepared by the National Committee for the Clinical Laboratory Studies, or NCCLS, that sets the standards for interpreting the zone size of bacteria when you're doing the Kirby-Bayer disc diffusion test. So for example, for the antibiotic penicillin and the organism Staphylococcus aureus, if the zone of inhibition is 20 millimeters or less in size, we say that this Staphylococcus aureus is resistant to penicillin. If the zone of inhibition is between 21 to 28 millimeters in size, then the organism is considered to be intermediate in its resistance to penicillin. And this is penicillin G, by the way. If, on the other hand, the zone of inhibition is 29 millimeters or greater in size, then we say that that bacteria is sensitive to this antibiotic. Okay? And as you come over here, let me blow this up. Here is our disc containing penicillin. That's why it's penicillin G, has the P there. And then you put the ruler down and you read this, and it looks like the size of the zone of inhibition is 20 or just a little bit less than 20 millimeters, in which case it is resistant to this antibiotic. Any question about any of that? All right. So the complication is you have to measure the size of the zone of inhibition and then compare that measurement to the chart to determine if the bacteria is resistant, intermediate, or sensitive to this antibiotic. You simply can't look, for example, and you can state that is that sensitive or resistant. You've got to look it up in a chart, and it depends on what the antibiotic is. I can't read them on this one here, and what the size of this is. Now, if there's no zone of inhibition, it's fairly safe to say that the bacteria is resistant to that antibiotic. And that one may be no zone of inhibition as well. Any questions about that? So on this one and that one, we don't know if it's resistant or sensitive because different antibiotics have a different size to be sensitive in their zone of inhibition to the antibiotic. And you just have to look. The larger antibiotics will have a smaller zone of inhibition than a smaller antibiotic. That's one thing that can affect it. And that is taken into consideration by the people who make this chart. Let me come down to the chart for Staphylococcus aureus. Okay. I think penicillin G is one of the smaller antibiotics, but I'm not 100% certain about that. All right, any questions about that? If not, know the terms of this lab and then work on the questions. Let me go to the questions. And that'll be in the worksheet, I think. Remember to only submit the worksheet. Do not submit the lab module. It looks like this worksheet's cut a little more. So let me go back to the module. So you're not physically going to perform any of these activities. This class is online, but you will be held responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bayer disc diffusion test. We have a little video for you to watch on preparing a spread plate to make the spread plate, and then review the steps for using aseptic technique for preparing a spread plate. And let me go through and talk about this now. So I'm going to make this my plate. I guess I can use this. And this will be my swab. So the swab has to be sterile. And then you dip it in the broth culture containing the bacteria, and the bacteria have to be well mixed. And then you get the worst of the liquid off on the side of the tube. So let's pretend this is my tube of bacteria. And you stick the swab in there and you touch the side. I don't really want to touch this because of my glass. I'll do it on the outside. You do it on the inside. And get the worst of the liquid off on the inside of the tube. And then you take the swab and then you inoculate the surface of the agar plate everywhere on the plate. It would be better if it was round so it shows a little better. You have to be careful because this has water in it. I don't know if you can see this, but you're swabbing everywhere. Okay? Can you see this? People? Can somebody answer? Hello?
I think if you hold it up a little bit higher, we could see it better.
Hold this up here higher? Is that better?
Yeah, that's better.
So you swab everywhere on the surface of the agar plate. And then once you're done it swabbing one time, you turn the swab a fourth of a turn, and then you rotate the plate a fourth of the turn. I don't know if you saw me rotating the plate. I didn't have it up high. And then you swab again everywhere. Okay? And then you rotate the swab a fourth of the turn, and you rotate the plate a fourth of the turn, and then you swab the plate everywhere once more time. The purpose is to get the bacteria growing everywhere. And then at the end, you take the swab and you go around the edge of the plate to make sure you get the edge, because when you're doing the streaking, you might miss the part of the edge. So you go around and do the plate. And that will get the bacteria everywhere, and the bacteria will grow everywhere, essentially confluently. And we call that a lawn of bacteria, because the bacteria will grow everywhere, sort of like green grass is everywhere in your lawn, assuming you have a nice lawn. Oops, sorry, I got the news coming up. You then put the cotton swab in the container, having biocide, label the plate, label on the bottom of the plate, and put the well, that tells you how to do the plate. Where is it telling you to put the antibiotic discs on? I don't see that listed here. All right, this is preparing the spread plate. And then you let the plate sit for 5 minutes, no longer than 15 minutes. And then you put on the antibiotic disc. That should be another step in here that's missing. And if you have a special tool, you can just put all of the antibiotic discs on the plate at one time, meaning it's something that has a lever and you push it down and it puts down all of the antibiotic discs all at one time, scattering them around the plate. And then it comes up and you remove it from the plate and you put on the lid. And then you let that sit just for a few minutes. And then you turn the plate upside down and put it in the incubator. Let's come to where we were. And let it incubate overnight for 35 degrees Celsius. And there is a specific time limit, which in the lab we almost never follow because it has to be 24 hours in the lab, unless somebody comes in to pull it off. But you should let it grow only 16 to 18 hours. And then if you need to, like in the lab, sometimes we will pull it, we can put it in the refrigerator, and then students, we take it out of the refrigerator, then students can look at it. We sometimes do that because most of the times our labs are 2 days apart. And then you look at it and you look at the pattern, the zone of inhibition. And you do have to measure the zone of inhibition with the ruler, measuring in millimeters. You can view the video clips and complete the virtual lab. Let's see if we actually have a virtual lab here. Come on. There we go. And then we have a virtual, nope, this uses Flash Player. So you have the virtual lab. Mine is a little older, that should be crossed out. I should look to make sure you don't have one online because I've got the new one online, but actually I've got it here too. Let me just look briefly here. The newer one. Down here. It looks like maybe I found another virtual lab at a different site. Let's see if this, yeah, we do. So we do have a virtual lab for this one. So click on that link for the virtual lab and you click on start. We'll get back to that. Let me get back to where I was. Where is it? Here it is. Nope, that's the worksheet. And then we have a little video for interpreting the results of the Kirby-Bayer disc diffusion test. So watch both of these videos. The virtual lab you have, if you especially have a slow internet connection, it may take a little while to download, but once it pops up, it should look like this. Click the start button. And then what you do is you take the ruler and you measure the diameter. And ideally, you should measure it 2 ways, meaning on this angle and across this way and this way. Of course, I can't change the ruler to get it to go that way. So we're only going to do it one time. And it's about there. And this one's about 3. That's 3 centimeters. So that would be 30 millimeters. And C is right here. And then you select which one it is. This would be resistant. That would be intermediate. And that would be the bacteria E. coli is resistant to C, which is chloramphenicol. And if it's green, it means you made the right choice. If you check the wrong one, it'll put a red X on it. And then you can try again if you want. Okay, so that's how you do this virtual lab. Any questions about that? Now go on the menu. There's different steps. It tells you about the procedure, which I've already gone over. Come on. Boy, that's not working. Not sure why that's not working. The different steps you use. Restart. And I thought there was another plate. This is, oh, there it is. This is a different plate here. This is, can anyone read that? Maybe Pseudomonas aeruginosa. And let's go and hit menu again. I guess hit restart. And this is Staphylococcus aureus. And this is a funny way to do it. Normally you can go through the different steps. But it looks like for this one, you just have to hit restart. And this is E. coli, hit restart. And then you can look at Pseudomonas aeruginosa. And that one has a zone of inhibition. That one has a very large zone of inhibition. For some reason, they're very white on this one, the zone of inhibition. And then this one is Staphylococcus aureus. And so measure those, and that would be your virtual lab. And like I said, it tells you if it's correct. It'll be green if it's correct. Whoops. Oh, this is a different bacteria, sorry. No idea, so we're just going to select. This one is resistant, 12 is green. All right, any questions about the virtual lab? So you do have a virtual lab, so I'm going to remove that. It's just I got to make a new link. Yep. And hopefully I'll get the new link in here. I'm not going to do it now. All right. So using the standards required for the Kirby biosusceptibility testing, an E. coli culture is prepared using a spread plate technique. Antibiotic discs are placed on the surface of the agar. Plates incubated 35, and below are the results, along with the NCCLS zone standards that you use for interpreting the Kirby-Bayer antibiotic test. When you're doing this, it's important that you measure the zone of inhibition, and it's important that you listen here, that you cannot measure it from here to here, meaning the zone of inhibition is not 10 millimeters, because if you notice, this 0 is not here. The 0 is there, and so how you read this is you read where this 0 is. So this zone of inhibition is 15 millimeters, okay? And you guys can read this here and here and here. This is E. coli for the zone of inhibition started or occurring with ampicillin, cephalophyllin, gentamicin, and tetracycline. And then you compare those measurements to the NCLS recommended standards. So for E. coli, for ampicillin, if the zone of inhibition is 13 millimeters or less, the E. coli is considered to be resistant to the ampicillin. If the, we'll just go up here. If the zone of inhibition is 17 millimeters or greater, then we say that the E. coli is sensitive to ampicillin. And then it's intermediate if in between. Okay, so you'll really only have to use this table because we're only looking at E. coli. And then we have the zone of inhibitions here. And so record the measurements of the zone of inhibition in this table here, and you can put it in, type it in directly like that. And then give the interpretation. So you go S for the ampicillin, E. coli is sensitive to ampicillin, or I if it's intermediate to ampicillin, or R if it's resistant. So put your interpretation and you'll be graded on your answers for this table. And are we in the worksheet now? No, we're not. Let's go ahead and move over to the worksheet. Let's close that down. So there's the zone of inhibitions and the table. And then there's the table you should fill in. And then answer question 3. This is question 2. Question 3 is, list the variables that must be standardized when performing an antibiotic susceptibility test using the Kirby-Bayer disk diffusion method. And remember, we standardize all these variables to make it so that different people in different parts of the world will have similar, if not the same, results using the same strain of bacteria and the same antibiotics. And you can see that you have to answer 1, 2, 3, 4, 5 different answers here. And you can get those answers in the lab module. I did actually talk about them when I went through the lab module. And then question 1, the size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? So write your answer. And then 2, what would happen if we didn't use aseptic technique to put the antibiotic disc on the plate? So write your answer. And you should explain it in terms of the Kirby-Bayer disk diffusion result. Meaning if you just tell me, if I don't use aseptic technique, I will get contamination. That is correct, but it's not talking about the Kirby-Bayer test. What will happen to the test? So tell me about that. What would happen if we increase or decrease the concentration of the antibiotic in the disc? So let's go back to, well, we would just use this. If we were to increase the gentamicin present in this disc, would that affect the zone of inhibition? If you don't remember, we did discuss that in the lab module. Sometimes students only answer this one partway. So they discuss what happens if we increase it. And if you only give one answer, that doesn't explain what happens if you decrease the concentration. So you have to answer for both, at least if you want full credit. And then 4, antibiotics can be bactericidal. They kill the microbe, or they can be bacteriostatic. They inhibit microbial growth, but do not kill the bacteria. When you're observing a zone of inhibition around the disc, how would you determine whether the antibiotic causing that zone of inhibition to be seen? How could you determine whether that antibiotic was bactericidal or bacteriostatic? This is a thinking question, and students often get it wrong, at least the first time. You have to think about this, and you have to think a little bit hard about it, okay? So I'll just warn you, you'll most likely get this one wrong the first time. Occasionally students get it right the first time, but oftentimes they get it wrong. So you have to think about that one. When performing the antibiotic sensitivity test, it's important that the test conditions are kept very standardized, like we do it using the same type of media. We have the same depth of the agar, the same age of the culture. And I think I mentioned that if you have a thinner depth of agar, the antibiotic will diffuse further, quicker and further than if you have a thicker depth of the agar. The age of the culture, new freshly growing cells will be more sensitive to the antibiotic than older cells. And that's partly because older cells are not dividing, or some of them are not dividing. And cells that are just being, I don't know, static are harder to kill than cells that are freshly growing. 6, are there other substances besides antibiotics that we could evaluate using this method? They're talking about the Kirby-Bauer disc diffusion method. And you should state what those other substances are. So don't just say yes or no, give an example. And you only need to give one, but you could give more. Does the presence of the zone of inhibition necessarily mean that the bacterium being tested is susceptible to that antibiotic? And that we discussed in the lab module. All right, any questions about this? If not, that's it for the lab. I will be here until 8 o'clock to answer questions. So get started on the lab. If I walk away from my desk, it's because I'm going to go get dinner, and I'll bet I'll eat it at my desk. So I will be here until the last student logs off or until 8 o'clock. When either of those two situations happen, I'm going to log off also.